Alright, this is part 1 to chapter 1, and for the remaining of the semesters, what you're going to find each week are going to be probably 2 to 4 short lectures. If you were in class taking this face-to-face, you would be in lecture for about 3 hours and interacting with the professor in that way. For the online component, what you're going to have are actually brief lectures and then a variety of different animations or additional videos to watch. So there could still be about 3 hours, 2 to 3 hours of short lectures or videos. And then you'll spend the rest of your time, just like a face-to-face student, studying and reading and completing assignments. So part 1 of chapter 1, this is really an introduction to microbiology as a whole, as the field. And here you have chapter 1 objectives. And so each chapter will begin, probably slide 2, of a list of objectives that you should be able to know after you finish this chapter. So you will use the list of chapter objectives to make a study guide. For your quizzes, that will be online on your own during the week. You can use your notes, you can use your books. But for your exams that you will take at the CWI testing centers, all of that is from memory. So what you need to do is take this list and create a study guide. The terms here, you've got a variety of different terms. Most of these terms are going to be bolded in the text. Maybe flashcards are a good option for you, but you will need to know what a pathogen is. What's the difference between a pathogen versus an opportunistic pathogen? Alright, so some of these are just basic definitions. Some of them are more of a concept, but as you can see, there's a fair amount of information here. All right, and then you go on to bigger ideas. All right, understand and know table 1.1. We'll look at that. These are individuals who contributed to even current day microbiology, how microbiology, the field of microbiology, started. All right, and so just some good basic information here. Now, it's important that you get a grasp on this information early because this is basic, and so the rest of the semester is going to build on chapter 1. Now, if you look through chapter 1, what you may have noticed is that this list of objectives actually only covers about half the chapter. That's because the second half of the chapter talks about microscopy and different stains and different techniques. You will do that in the lab. And so I'm not going to go over that material in the lecture. Alright, so what is microbiology? Basic term, definition. Microbiology is the study of microorganisms or microbes. You'll hear some of those terms used interchangeably, microbes, microorganisms, and then you can get into more specifics in terms of what microbe encompasses. All right, cellular living microorganisms such as bacteria, archaea, fungi, protists, helminths. Most of these are going to be invisible to the naked eye, but that's not all cases. All right, there's plenty of fungi out there that you can see. Mushrooms are macroscopic fungi. Helminths are parasitic worms, and some of these can actually be quite large. All right, but some of them are going to be small. All right, then there's non-living, non-cellular entities such as viruses, prions. Those you cannot see with the naked eye. Those are not alive. Viruses are non-living. They are not a cell, but rather they are an entity. And with viruses, the proper terminology is to talk about them being active or inactive. An inactivated virus can't infect. An active virus could infect a cell if it had the opportunity. All right, so table 1.1, living and non-living agents studied in microbiology. So big picture here, and looking specifically at the microbes, whether it's bacteria, the archaea, protists, fungi, helminths, viruses, prions, and the cell type. A major way to differentiate between cells is whether or not they are prokaryotic or eukaryotic. Eukaryotic cells are like us. We are eukaryotic organisms. All of our cells, except for red blood cells, have a nucleus. Prokaryotic cells do not have a nucleus, and that's one of the major ways to divide cells between eukaryotic versus prokaryotic. All right. Bacteria are considered unicellular, so that it's one cell. They can be pathogenic or non-pathogenic, meaning they can cause disease or they don't cause disease. The archaea are very similar to bacteria. There are some differences, but typically what archaea are thought to be as ancient bacteria because they can live in extreme environments, whether that is sometimes a very high salt content, high temperature, high pH, low pH. They're the extremes, and they are sometimes referred to as extremophils. All right, the rest of the group, protists, fungi, helminths, viruses, prions, we'll talk about those in additional chapters as we move through this semester. But for right now, what you do want to know is the cell type. So if you were to be given a matching table, you could match this group, right, the individual type of microbe, with whether or not they are eukaryotic, prokaryotic, or non-living, so to speak. Okay, so brief description here. Prokaryotic cells, excuse me, considered to have evolved about 3.5 billion years ago, earliest forms of life, and they include the bacteria and the archaea. All right, eukaryotic cells are composed multicellular organisms. All right, and there's also unicellular. So eukaryotic cells can be both unicellular and multicellular. And then there is the endosymbiotic theory, which that is not something that is put into or found in this chapter, but endosymbiotic theory is really an idea of how eukaryotic cells could have evolved, with there being a large prokaryotic cell and a small prokaryotic cell. And the large prokaryotic cell, so two bacteria, one big, one little. The big bacteria cell engulfs the little bacteria cell, and then that little bacteria cell becomes part of the big cell, and that is, in theory, how the nucleus developed in the eukaryotic cell. So, endosymbiotic theory, not necessarily the nucleus, the mitochondria. How the mitochondria are important for energy production is referred to as the powerhouse. And prokaryotic cells do not have mitochondria. Eukaryotic cells do, so another difference between those two groups. And endosymbiotic theory is how eukaryotic cells developed mitochondria. All right, so microbes and disease. All right, part of this field is about the basics of understanding health and disease. Pathogens are microbes that cause disease. All right, about 1,400 pathogens are known to infect, and those pathogens can be viruses, bacteria, fungi, you name it. But we do understand, we're pretty sure that less than 1% of all microbes are pathogenic. And that's actually probably a very small number because we know that there are thousands, if not millions, of different microbes that are out there. It's estimated that we only are aware of about 1% of the microbes that are out there. Why? Because they don't do anything, or they don't do anything that we pay attention to. There are large numbers of bacteria and fungi that are in the ground. Our planet, human beings, animals of all kinds, would not be able to survive if it weren't for the bacteria and the fungi that are part of our normal flora. Anyway, opportunistic pathogens are pathogens, but they only cause disease in a weakened host, or if they get into the wrong area. So, for example, E. coli is a major component of our bacteria in our gut, in our intestinal tract. And in the intestinal tract, the normal E. coli don't cause problem. In fact, they are extremely beneficial to us. However, if that same bacteria, that same E. coli, moves from the intestinal tract and makes its way into the urinary tract system, well, now the person can develop a urinary tract infection and cause problems, cause discomfort, cause issues. All right, so that's an example of an opportunistic pathogen. In terms of a weakened host, we can use, there's a lot of examples of that, but we'll go with someone who has AIDS, all right? A person with AIDS, they virtually have no immune system. Their adaptive immune system is being wiped out because of the presence of the HIV. And Candida albicans, again, a normal flora for humans, is able to expand in high numbers and actually cause disease in AIDS patients. And though that what seemingly would be a very benign infection in someone who had a competent immune system, you probably wouldn't even get a systemic yeast infection if your immune system was competent. But with an AIDS patient, Candida albicans can actually kill a person. All right, figure 1.1, you don't need to pay too much attention about this, but just recognize that there's what's called the golden age of microbiology, just basic understanding, Jeopardy-like questions. When during this 50 years or so, the end of the 1800s, very beginning of the 1900s, there was a lot of work being done in this process of trying to understand really human disease, what causes disease, and try to improve human health. All right, spontaneous generation versus biogenesis. All right, life evolves from itself, or there is a life form where life comes from life. Some of these individuals are very important in microbiology. There's Robert Hooke, and Robert Hooke was the first to publish a description of cells. And both he and Leeuwenhoek, all right, talked about animalcules. All right, little tiny animals that were more like molecules. And both of these individuals were very important in establishing really the desire to know more about the world around us. Leeuwenhoek is considered to be the father of microscopy. He was the first to see bacteria. He made his own microscopes. They were very primitive. You see this, and it really doesn't look like anything. Right? Not a microscope that you would think was a microscope. But then he drew these drawings. All right, and this was the first indicator that microbes were different from each other and that they looked different. They had different forms. All right, so again, with increasing the ability to observe cells, the debate of origin of life came about. And there's the idea of spontaneous generation that was very popular in the 1600s and 1700s. Life comes from non-living items. And then biogenesis, life emerges from existing life. And this was a hot piece in both science as well as religion at the time. And this is where we'll stop part 1. We'll continue with spontaneous generation, but a different twist on that.